Welcome, students. I am Dr. Seema Desai Fernandez, Assistant Professor, Department of Botany, St. Xavier's College, Mapsa, Goa. This program is for TY BSc Botany student, Semester 5, paper code BOC 106. Title of the unit, Unit 3 Cell Division, Module name Mitosis, Module number 27. Outline, Mitosis cell division, and you are going to learn types of phases. Learning outcomes describes the mitosis cell division, explains the types of phases. Introduction. What is mitosis? Mitosis is a type of cell division seen in eukaryotes. In this process, a parent cell divides to form two daughter cells, which are genetically identical to parent cell. The phase at which a cell prepares for division is called interphase. Mitosis is also known as equational division because the number of the chromosomes will remain same in the daughter cell as present in the parent cell. It is a phase where nuclear division takes place, and this is known as karyokinesis. It is divided into following phases: prophase, metaphase, anaphase, telophase, and this is accompanied by cytokinesis. This is the figure which explains the mitosis cycle. The cycle starts with the interphase G1, S phase, and G2 phase. In G2 phase, the cell prepares itself to go into the mitotic cell division and starts with the first phase, that is the prophase, second phase metaphase, anaphase, telophase, and then the telophase is accompanied by the cytokinesis, where the cell divides into two new daughter cells. Some cells goes into the resting phase, that is the G0 phase. This diagram shows all the phases of mitotic cell division cycle, starting with the interphase, prophase, prometaphase, metaphase, anaphase, and the last phase of the mitotic cell division is the telophase, which is accompanied by the cytokinesis. These cells, each cells are magnified in the next slides. The first phase is the interphase. Interphase is the longest part of the cell cycle, during which a cell increases in size and produces organelles, which will be divided between two daughter cells. The amount of DNA is doubled during this period. This is the figure shows the interphase where the chromosomes are in the uncondensed form. Just before the next cell division, the chromosomes replicate so that each then consists of two chromatids joined together by the centromere. There is considerable metabolic activity as these processes need energy in the form of ATP. The chromosomes are not visible at interphase because the chromosome material chromatin is dispersed throughout the nucleus. It is only during the first phase of mitosis, called prophase, that the chromosomes start to condense and become visible. The first phase of mitotic cell division is prophase. During this prophase, chromatin condenses to form chromosomes. There are two identical copies of each chromosome, which are attached to centromere. During the end of the prophase, nucleolus dissolves. This figure shows the nucleolus is disappear. This figure shows the nucleolus has disappeared and the chromatin is showing two sister chromatids. Nuclear membrane also disintegrates in this phase at the end. The centrosome moves to the opposite poles. Spindle fibers starts to appear. The appearance of the spindle fibers in prophase 1 the appearance of the spindle fibers in prophase is very important phase in this cell cycle. Brief explanation on the formation of spindle fibers in plant cells. Spindle fibers compose a protein structure segregating the genetic material in a cell. It consists of fibers wherein the replicated chromosomes link at their centromeres. These spindle fibers are later functional in pulling apart the sister chromatids to the opposite poles, so that eventually the two new daughter cells that have formed will each possess a set of chromosomes. Plant cell lack centrioles, but still they are capable of forming a mitotic spindle from the centrosome area of the cell located just exterior to the nuclear envelope. The next phase is the pro-metaphase. In this phase, the replicated chromosomes migrate to the equatorial plane. In the midline of the cell, the mitosis spindle region is called the metaphase plate. The figure shows that the chromosomes migrated to the equatorial plane, that is the midline of the cell. The next phase is the metaphase, in which the chromosomes are condensed and the chromosomes align themselves in the equatorial plane, that is on the equatorial plate, also called as the metaphase plate. The analysis of the metaphase chromosome is used in cytogenetics and cancer studies. The figure shows that the chromosomes align themselves on the equatorial plate, that is the metaphase plate. The next phase is the anaphase. It is the shortest and the rapid stage of the cell cycle. In this phase, the replicated chromosomes move apart by the centromeres and daughter chromatids moves to the opposite poles as the kinetochore microtubules shorten. The figure shows that the chromosomes are pulled towards the opposite poles and the kinetochore which is attached to the chromosomes are becoming shorter. Chromosomes are maximally condensed during the late anaphase. The chromosomes appear in anaphase, the Y-shaped, as they move to the opposite poles. So here the picture is showing the chromosomes are in the form of Y shape. The last phase of the mitosis is the telophase. Chromosomes arrive at the opposite poles and begin to condense. Chromosomes arrive at the opposite poles and begin to decondense and lengthen. Nuclear envelope reappears and surrounds each set of chromosome. This is the figure which shows the chromosomes are in the opposite. This figure shows that the chromosomes are at the opposite poles and the nuclear envelope reappeared and surrounds the sets of chromosomes. And the chromosomes begin to decondense and lengthen. Telophase is accompanied with the cytokinesis. In this phase, two daughter nuclei are formed. Spindle fibers continue to push poles apart. The mitotic spindle breaks down. The late telophase is marked by the cytokinesis. This is the figure which shows that there is a cleavage furrow formation and the nuclear envelope is forming back and enclosing the chromosomes. The cell is undergoing the process of the cell division into two daughter cells. Cytokinesis is the division of cytoplasm, organelles, and cell membrane to form two cells. It divides the cell into two daughter cells, which are identical to the parent cell. Animal cells shows a cleavage furrow which separates the daughter cells, whereas the plant cells shows a cell plate forms across the equator of the parent cell from the center outwards and a new cell wall is laid down. Significance of the mitosis. Mitosis produces two cells that have the same number of the chromosomes as the parent cell, which are haploid, and each chromosome is genetically identical to its original parent cell chromosome. Therefore, the division allows the production of the cells that are genetically identical to the parent and so gives genetic stability. By producing new cells, mitosis leads to growth of an organism and allows for repair of tissues and replacement of the dead cells. An example of mitosis in plants is in the root tip. In human skin, dead surface cells are replaced by identical cells from below. Asexual reproduction results in complete offspring that are identical to the parent. Asexual reproduction involving mitosis takes place in certain flowering plants where organs such as bulbs, tubers, and runners produce large number of identical offspring in a relatively shorter period of time. There is no genetic variation between each individual. However, most of these plants also reproduce sexually. These are the references. Thank you, students.